All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So this talk has been asked a lot, I think even over for a year, and that is what is the role of MTHFR enzyme in clotting in context of the COVID or vaccine and in general as well. So that is what we are going to talk about. In today's talk, we are also going to talk about this little tongue and groove plier set or plier as well. And we'll talk about these beautiful little spinach leaves as well. So these are our characters today. And this spinach is mine, so I will I will eat it afterwards. So with the leaves representing folic acid and with the pliers representing enzymes, let's start our discussion. Very important discussion in general for cardiovascular health, and then especially in the case of COVID and vaccine as well. So this is the another gift for humanity, and these are continuing. Just very quick references. This is one of the articles about MTHFR, homocysteine and MTHFR mutations. Then this is another paper. All of these links are present in the description. Then here is one more, cardiovascular diseases and MTHFR gene. So if you see here, we'll talk about this as well. Then here is the folate insufficiency due to MTHFR deficiency. We will talk about it. And then there are some more readings for you if you would like to. I will do some more discussions later on in this series too. Today I'm going to build the foundations. And finally, this is drbeen.com. I have the good news that the courses that I was working on, remember the descriptions were left. So the writer has actually written all the descriptions. So we are good with that. Now only the prices are going to be adjusted and we will transition to the courses. So that is another good news that this is finally done. Prices are still, they need to be taken care of. But if you go to the Dr. Been members or drbeen.com/courses, you can see various lectures, their courses, their CMEs, and so on. Now, in the meantime, we have also launched a new 3D anatomy system that is in beta and we are adding to it. But for example, if you go here, let's say you want to read about tibia, you would be happy that we have written an anatomy book. So as you can tell that these are still broken, so we're working on it. But this is the 3D anatomy module that has become available. And you can click on any of these terms here. For example, if I click on femur, it will go in this 3D anatomy module and show you the femur. So these are all live link with the 3D anatomy. This is also part of these plans. So in the description of this video, there is a link which you can see the price here. I'm not even going to speak the price. It is so low. This is a one-time price, no further fees, and that gives you access. What I'm going to do is, because we are ready with the courses now, I'm going to leave this week, maybe another 5-6 days with this price. Then we'll change the price to our regular price, which is $97. And then we would just altogether take away the lifetime and just switch to the courses. So take advantage of this if you would like to. So with this, let's start with my drawings. So methylene tetrahydrofolate reductase enzyme deficiency and cardiovascular health. The cardiovascular health over here, again, related to the vessels, which in turn is also related to clotting. And now keep in mind that the important takeaway is going to be that the individuals with MTHFR gene deficiency or enzyme deficiency can not gene mutation and enzyme deficiency can end up not being able to metabolize folate and methylation pathways very successfully, resulting in reduced methylation processes in our body, which can actually impact hundreds of chemical reactions and enzymes, which can impact many parts of our body and its functions, including endothelial uh, disruption, endothelial function disruption, clotting factors disruption, and so on. So that is the basic gist of this talk. So we're going to start from here and think about it. If somebody is already MTHFR, has an MTHFR gene mutation, and has a propensity for clotting, and on top of that, if they become COVID positive, COVID also increases the risk of clotting. Vaccines in some people can increase the risk of clotting. So imagine that additional risk that can be posed. And this can be posed by anything that would increase the risk of clotting when the person also has mutations of MTHFR gene. So here, let's start with this one. It's really a cute little cartoon story. So imagine we have folate or folic acid or vitamin B9. It is a water-soluble micronutrient or vitamin. Normally it is found, I mean, it is in many, many things, but green leafy vegetables, bananas, avocados, legumes, the various kind of nuts, eggs, various meats, especially liver, spleen. So folic acid is ingested by us. Now what happens is in our body, folic acid, here, this little leaf is combined with water. So these two little rounded things are two waters. So it becomes dihydrofolate. So from folate, we make dihydrofolate. Then we add two more waters and we make it tetrahydrofolate. I know that I could have actually put the actual equations here, but those become too boring. So let's do this little cartoon thing. So we get folate, we make dihydro, two hydros, then four hydros, tetrahydrofolate. Then what we do is, with the tetrahydrofolate, we add two methyl groups. Methyls are CH3. So methyl groups or methyl groups, whichever way you pronounce it. So if you see here, 5,10, so that is methyl group attached to the carbon number 5 and carbon number 10. And then the remaining structure is tetrahydrofolate. So now we have 5,10 tetrahydrofolate. There are two methyls. Then look at this guy. This is the MTHFR. So MTHFR, methylene tetrahydrofolate reductase, it is an enzyme. It reduces tetrahydrofolate. So if you see here, what it does is this enzyme steals one of the methyl group from the tetrahydrofolate, which is methylated. So then we are left with, on this side, if you see this folate is now crying. Why? Because MTHFR has stolen a methyl group from it. And so now this one is left with 5 tetrahydrofolate final shape. The 10, the methyl group on the 10th position has been taken away. This is the function of the MTHFR. So MTHFR enzyme is going to be able to help accelerate the methylation processes or reduction processes of the folate pathways. So now, keep an eye on this guy. What is this guy? This guy has 4 hydros, so tetrahydrofolate, but it only has 1 methyl group. Now, if we steal this remaining methyl group as well, then this little structure will become tetrahydrofolate, right? So that is the difference between them. 5, 5, methyl, So here methyl tetrahydrofolate, if you take this methyl from it, it will then become just simple tetrahydrofolate. So that is what happens. This tetrahydrofolate, it goes between these two states. Tetrahydrofolate becomes 510 tetrahydrofolate, then MTHFR enzyme comes in and steals a methyl from it, then the remaining one methyl, and then tetrahydrofolate is here, then we steal the next methyl from it as well, and this little poor thing becomes tetrahydrofolate again. Good. Now the question is, if we steal this methyl from it, it's all about taking this methyl away from it. Why these little spinach things, these little folate things are helping us transfer methyl. The whole story is about this little methyl group, not about the folic acid or folate. This methyl group is then donated to many other enzymes that are performing various functions in our body. So these structures, these molecules here are really the carriers of methyl, and they're bringing these methyls to their destinations. And you can see here, before I move forward in this process, you can see here that if MTHFR isn't doing its job correctly, then this whole process is going to get impacted and slowed down and become inefficient. So keep that in mind. So here we have the 5-tetrahydrofolate, and that to steal a methyl. How do you take that methyl from it? Here is what you do. There is another molecule, homocysteine. What you do is you take, homocysteine is an amino acid. So what you do is you take this methyl from the tetrahydrofolate and you give it to homocysteine. So if you see here, this is homocysteine, and you take this methyl and attach it here. So that is what you see here. This is actually the same formula as homocysteine. The only thing is here we have this methyl group that we have taken from 5-tetrahydrofolate. So when we take that methyl group again, the tetrahydrofolate becomes tetrahydrofolate, and the methyl group comes in here, and now we have methionine. Good. So I'm going to reduce the size a little just to show you. We started from the folate. We made dihydrofolate. We put 2 waters on it. So if I take this little spinach leaf and I attach to water, I dip it in water twice, 2 waters. Then I dip it twice more, 4 waters, tetrahydrofolate. Then we attach 2 methyl groups to it. And now we have 510 tetrahydrofolate. Then the MTHFR enzyme comes in and steals one of the methyl, and we are left with 5-tetrahydrofolate. Then homocysteine comes in and steals another methyl group on it, and the tetrahydrofolate just becomes simple tetrahydrofolate. Homocysteine becomes methionine. Good. Now this methionine would then go back becoming homocysteine. And guess how will it do that? By losing this methyl group. It's all about the methyl group. These molecules have made a line, an assembly line, to hand over the methyl group from one to the next, to the next, to the next. So the methionine will then become S-adenosyl methionine. This would have the methyl group with it, and this methyl group will be then given to whoever wants it. And a lot of chemical functions in our body require presence of a methyl group. So methionine will become S-adenosyl methionine. Then this methyl group will be given to this little X, whatever X it is. And of course, that X product will now have the methyl, and then the adenosyl methionine will become adenosyl homocysteine. From there, it will become homocysteine again, and then that homocysteine molecule will pick up another methyl, and this continuous cycle would keep on going. What will be the result of this cycle? The result of this cycle is there are things that are receiving methyl groups. If you said that, man, all right, fine, we have these methyl groups like this little treasure present, What are we going to do with it? Many enzymes that are working to make DNA, that are working to make RNA, that are working to make proteins, that are working to make lipids, or are part of the metabolisms of these structures. These enzymes need methyl groups, or they work with the methyl groups. This is called methylation. We do methylations of a lot of molecules in our body when we are performing various functions. And for that methylations, we need enzymes that will do the methylation, but we also need methyl. And if we don't have methyl, or if we have less methyl, then we have a problem. So then, what kind of a problem can occur before we go into the genetics of this? You saw that the folate here, there are hundreds of functions in our body where methylation is done as part of the enzymatic action. That methylation, that methyl group, is brought to those enzymes through the little assembly, little machine that I showed you over here. Just have a quick look at it. This is that little machine. The beginning from the folate, ending to whatever action which needs the methyl. And this methyl group is just tossing around through this whole thing and going here. And this guy, methyl tetrahydrofolate, is sitting right in the beginning. And in some people, there is a deficiency of this enzyme, or there is an abnormality of this enzyme, mutation of this enzyme. The structure isn't as normal in other people. And so this enzyme performs lower, it performs lesser than others. And that reduced performance causes less methyls to be available, causes this machine to run slowly, and we get pathologies. And I'll show you what those pathologies are. I mean, some of those. Of course, I cannot. There are hundreds of functions, so some of them. So now that we know the importance of MTHFR enzyme, let's look at how this enzyme is built. So on our chromosome number 1, there is a location on the petite arm, or the small arm of the chromosome. So chromosomes have a small arm and a long arm, or Q and P arm. Petite arm, location number 36.3. So we'll read it as 1, chromosome 1, P36.3. That is where the gene is located that helps make MTHFR enzyme. There are 2 genes, one from father and one from mother. Now, in some people, if we go here, in some people, if you see here, one of the genes has a mutation on it. There are found to be 24 mutations in total. But let's say in some people, one of the genes has mutations, other gene is normal. This is called heterozygous. Why? Because they both are different from each other. This is heterozygous activity. If they both are similar, that will be homozygous. This is heterozygous. Then in homozygous, if you see here, both genes have a mutation, similar mutation, but both of them have that mutation. This will be called homozygous mutation or compound homozygous mutation. Then another possibility is that the gene coming from mother and father, they both have mutations, and it's not necessary that mother or father gave the mutation. Mutations can occur after we receive the genes. So, and mutations can occur during our lifetime as well. So, but the mutations are different. They both are mutated, but one has a different mutation and the other one has a different mutation. This kind of a mutation is called double heterozygous. Heterozygous because they're different from each other, but they both are mutated. That is called double heterozygous. So we have all those mutations that are possible on the MTHFR gene. Now the question is, what are the chances that we have those mutations? So here, epidemiology, there are, as I said, there are 24 mutations. This is the most common C677T mutation is the most common mutation. This mutation in the US in whites and Hispanics, 20 to 40% of the population has this mutation. Check this out. 20 to 40% of whites and Hispanics in the US have this mutation. And this is a heterozygous mutation. That means one allele has a mutation that causes the resultant MTHFR enzyme that is produced to be 65% of the normal performance. And this is where I wanted to bring in this little plier, tongue and groove plier. So look, imagine if this plier is an enzyme. You can see that I can actually open it incorrectly, and now the function will become impacted or different. So imagine if I am trying to really hold a very tiny thing, let's say this pen in it. It cannot hold that pen at all because the shape is changed. I can change the shape and make it hold it better, or I can change the shape to make it hold it loose. So now it is loose. And so the enzymes function is to increase the rate of chemical reaction without getting consumed in that reaction or becoming permanently altered. The second part of the enzyme's function is to not throw the balance of the substrates and the resultant outcomes out. So keep the balance as well. So that is a function of the enzyme. Now, if I go back to this place here, this mutation, which is present in 20 to 40% of whites and Hispanics, causes their enzyme, MTHFR enzyme, to be 65% of normal. That means if I eat the same folate amounts and another person of a similar body habitus, similar body needs, takes the same amount of folate, then their enzyme will only help with 65% of the methylation function or reduction function. It is methylene tetrahydrofolate reductase. So reduction function. In US, in blacks, this mutation is only 1 to 2%. So it is more common in whites and Hispanics. That means that potential risk or propensity or increased likelihood of developing clotting is more possible in whites and Hispanics compared to blacks. And then if you add COVID or vaccine, you can then see which population becomes more at risk, which tells me that it is really important for all of us to know if we have any mutations in this enzyme. It is important for us to know that. Now, in North America, Europe, Australia, this mutation is present in 8 to 20% of the population, and they actually have two mutations. So that is a homozygous mutation, and that causes the enzyme to develop even more performance issues, and it is 30% of the normal performance. So in North America, Europe, Australia, 8 to 20% of the population may have MTHFR enzyme performing only at 30% of the normal performance. Then another mutation is called A1298C mutation. This mutation is present in North America, European, Australian population, 7 to 12%. In Hispanic population, 4 to 5%. In Chinese population, 1 to 4%. In Asian population, 1 to 4%. And this also reduces the enzyme's function. So you can see that anywhere from 1 to 4% up to, in whites and Hispanics, 20 to 40% of the MTHFR enzyme performance issues can be seen. So then the question becomes, so what? What will happen? So we actually, because methylation is part of so many chemical functions in our body, that we actually do not very clearly know which enzymes aren't working correctly, where the methylation isn't working correctly because we do not have sufficient methyl groups available. So to pinpoint the outcome isn't really easy. However, pathologies are observed. And I would show you a American Heart Association's article where they say that there is nothing conclusive in this area. And it may even be that it's not the MTHFR enzyme. Instead, it is some other pathology, and MTHFR enzyme deficiency seems to be a marker of that pathology. So that means the confusion is there. However, in these pathologies, vitamin B6, vitamin B9, which is folate, and vitamin B12 really help. So if you have a takeaway from this whole discussion, vitamin B6, 9, and 12 are important to make sure that the clotting is not rampant. At the same time, if you have normal MTHFR levels, if your MTHFR levels, folate levels, and methyl levels, if your MTHFR is correct in its performance, then over adding the folate can also have symptoms. So this is not a vitamin that you can just overload your body with. So be careful that if you don't have this issue of MTHFR, then don't take extra. But if you have a problem with MTHFR performance, then taking the corrected amount is important of amount of folate. So B6, B9, and B12 can be given. So what are the possible pathologies in the clotting context? Number one, because of the lack or reduced performance of MTHFR and resulting less methylation actions, the endothelium of blood vessels can become damaged, dysfunctional instead of damaged. The endothelium of the blood vessels can become dysfunctional, resulting in inflammation and reduced production of nitric oxides and other molecules by the endothelium, which those molecules, we have done this discussion before as well, endothelium produces molecules that help prevent clotting in a normal healthy blood vessel. Now, if the endothelium is not working correctly, then within that blood vessel, clotting would start occurring. Endothelial function impairment will lead towards clotting, number one. Number two, the clotting factors. I made these clotting factors like little corks. So corks, because clotting factors help with clotting or putting a cork where the damage is. So clotting factors themselves can be impaired. Why? Because once again, for these factors to be produced, we need MTHFR enzyme and methylation activity. And when these factors are working, we will need methylations. So it is possible that in the folks who have MTHFR deficiency, they may have their clotting factors not working correctly or working abnormally, which would then also cause propensity towards clotting. And then there is also a possibility that as the cells are not able to perform their functions correctly, their enzymes are not working correctly, their methylation functions are not happening correctly, the reactive oxygen species that are produced may not be neutralized properly. And these extra reactive oxygen species will cause damage to the endothelium and other parts of our tissues. And if it is endothelium and heart, then once again, within the endothelium, we'll start getting clotting. So clotting because of reactive oxygen species, clotting because of the abnormal clotting factors, clotting because of the impaired endothelial function. And there are many other possibilities as well. The end result is cardiovascular health can become different or impacted. So that if we now add COVID, for example, on top of it, so if I go here, you have this little study here. Here, prognostic genetic markers for thrombosis in COVID-19 patients, a focused analysis on D-dimer, homocysteine, and thromboembolism. So if somebody has the MTHFR gene deficiency, have a problem with homocysteine, homocysteine is a marker. So if I go back here to this diagram, homocysteine here, this is homocysteine. If homocysteine is not able to become methionine easily, because these functions are not occurring and methyl groups are not available, then the homocysteine level will increase. So increased homocysteine level have their own pathologies, but homocysteine level increase is a marker as well to say that there is something going on. So this is one study. Similarly, we know that with the vaccine, if there is spike protein that causes binding with the endothelium and causes endothelial disruption and causes clotting propensity or increased likelihood of clotting or microthrombi formation, and if the patient already have or the person already have MTHFR enzyme deficiency, then they are at increased risk. So this is the last piece that I want to show you. This is 2015 American Heart Association. And what I want to show you is that for the majority of the pathologies, so what are the risks for someone with elevated homocysteine, which is in turn a marker, let's say, for the MTHFR deficiency? They say the cardiovascular disease, elevated homocysteine levels are associated with an increased risk of cardiovascular disease. The higher the level, the higher the risk. But then they say, in 2010, the American Heart Association issued a statement that it does not consider high homocysteine levels in the blood to be a major risk factor for cardiovascular disease. What they're saying is just a higher level of homocysteine is not, there is not sufficient evidence to say that there will be enough methylation issues that the endothelial dysfunction or the clotting factor dysfunction or reactive oxygen species attacks will be sufficient to cause issues. However, there is an association seen, there is no causation that is seen there. Similarly, clots in veins. Elevated homocysteine levels are associated with an increased risk for blood clots in the veins, and they talk about them. And then they say, however, overall, the risk of DVT, deep vein thrombosis, and PE, pulmonary embolism, is only slightly increased. And although elevated homocysteine is a risk factor for a first episode of DVT or PE, it does not predict a higher risk of recurrent clot once a patient is off blood thinners. Therefore, finding an elevated homocysteine does not influence the duration of patient should be treated with blood thinners. So that's also, yes, we see it, but it's really very light and no, you don't even have to give blood thinners. So that is, I wanted you to see where American Heart Association stands. Then same thing in the pregnancy, that they do observe elevated homocysteine levels have been observed more frequently among women with certain pregnancy complications. But if you then see here, however, it appears that the elevated homocysteine levels may be a consequence of these complications rather than the cause. And then they say it is recommended that all women of childbearing age take a multivitamin containing 0.4 milligram of folic acid per day to reduce the chance of neural tube defects in their children. So you can see that they are everywhere. So now here, how does elevated homocysteine cause cardiovascular disease? It is not clear whether elevated homocysteine causes the blood to clot more easily or whether it is just a marker for an increased clotting risk. Then they talk about it, and finally here in purple they say, accordingly, the American Heart Association in 2010 stated that a causal link between homocysteine levels and atherosclerosis has not been established. But at the end of the day, can homocysteine levels be lowered? Yes, they're saying use folic acid, which is vitamin B9, B6, and B12. So B6, 9, 12. So correct. LL Coolbein says it's a biomarker. So homocysteine itself is a biomarker to say that there can be many reasons for homocysteine to be increased. MTHFR gene and MTHFR enzyme deficiency can be one of them. If there is an MTHFR deficiency and generally methylation actions are not occurring correctly, then there can be an increased likelihood of clotting. So this is the discussion. Please like, subscribe, and share. This was a topic that had been asked for years now during the COVID time, and I am so late in it that I'm presenting it now. If you would like to dig deeper in it, please let me know in the comments, and I will do further discussions. But individuals with the MTHFR gene deficiency, gene mutation and the enzyme deficiency should discuss with their doctors and understand what is the risk of clotting, because that risk can become modulated when they get COVID or vaccines. So thank you very much. Like, subscribe, and share. And if you would like to support this work, if you like this work and you would like to support it, you can become part of the Substack or Patreon, or you can use PayPal, or you can buy me a coffee, or you can just become member of Dr. Bean. All the links are in the description. Now, tomorrow, as usual, I would have two talks, 1 p.m. Zoom and 6 p.m. Pacific time Zoom, only with the supporting members, YouTube channel supporters or Substack supporters or Patreon supporters will do the Zoom tomorrow at 1 and 6, and we'll have our little live talk then. Thank you very much, and I would see you tomorrow. I will not be live here on YouTube. Tomorrow also I'll be with the FLCCC as well. So their weekly discussion, I'll be presenting with Dr. Paul Marek. So thank you very much. See some of you tomorrow. Bye for now.